Cannabidiol, commonly referred to as CBD, is one of 113 identified cannabinoids that are found in cannabis. It, alongside tetrahydrocannabinol, also known as THC, each make up for about 40% of the substance. In a growing realm of legal medical cannabis use, you may have heard of these molecules more often as various types of cannabis and their different medical effects are being explored, separated, and categorized. Notably, THC is classified as being intoxicating and psychoactive alongside its psychedelic and euphoric effects, while CBD has a set of pain-repressing and anxiety-relieving effects that are non-intoxicating. Quite infamously, New England Patriots tight end Rob Gronkowski retired from football recently, 2019, saying that his body had been under so much pressure and pain over years of physically enduring football and 9 different surgeries that he had lost the joy in life. But after partnering with medical cannabis firm CBD Medical, apparently that issue of losing the joy in life was suddenly solved. That's real interesting. Okay, no, but seriously, I feel like if Gronk had tried traditional cannabis with THC, he may have just permanently gone into a retirement lifestyle of enjoying euphoric and psychoactive effects of traditional cannabis while getting out of shape and eating a lot. But CBD Medical's subtle line of CBD creams, oils, and nasal sprays may have given him non-psychoactive chronic pain healing and anxiety relief without too many side effects, allowing him to stay productive, and in turn he eventually returned to football physically and mentally relieved and healed, and even won another Super Bowl. So it is apparent that the isolation and specific use of CBD over other forms of cannabis consumption can be very useful for those looking for natural medication healing, hesitant to use opioid or amphetamine-related substances with addictive and damaging properties, but who also want to stay away from the high effects that many traditional forms of cannabis give out. So some of the possible positive effects of CBD include pain relief, anxiety and depression relief, potential cancer alleviating properties, acne reduction, neuroprotective properties, neuroprotection being the preservation of neuronal integrity as your neurons degrade over time, and heart health benefits. Now, obviously, excessive use of this molecule still has some side effects, as almost everything used in excess has side effects, but there seems to be appetite changes and fatigue. So for a brief research history of CBD, the cannabis plant was used historically for centuries, dating back to 500 BC, including Asian herbal medicine practices with cannabis and hemp plants. Hemp being a cannabis plant just with a lower THC percentage, so lower than 0.3 THC. Practices using these plants can be found historically throughout several cultures and even religions, but one of the first formal medical studies was done in 1893. Irish physician and medical researcher William B. O'Shaughnessy explored the rudimentary effects of cannabis and thoroughly described its potent medical applications, particularly as an anesthetic. Although William was an Irish physician, he was conducting these studies in India and was studying a natural hemp that was grown on the Hindu Kush mountains. And this natural hemp did not contain much THC at all, meaning that it was mostly comprised of CBD, which is why he found the anesthetic and pain-relieving effects in his study, rather than any intoxicating effects. And this is why hemp is used much more in modern CBD extraction, because it is composed of a higher amount of CBD rather than other cannabis plants, and thus gives more yield. And the molecule is exactly the same in both types of plants in terms of structure and properties. It just has a varying amount. In 1940, Robert S. Cann discovered and found the structure of the first cannabinoid, CBN, and 2 years later, Roger Adams successfully isolated the first ever cannabinoid, which happened to be CBD. With some very deep searching, I was actually able to find the paper that Roger Adams wrote in 1942, where he referenced Robert S. Cann's work in 1940 and expanded upon it with his findings on CBD so far. He wasn't even 100% sure of the structure, not sure where a possible double bond or two may have been located in the structure, as he knew there were possible double bonds based on the formula they had discovered, looking at the ratio between hydrogens and carbons based on the structure they had known so far, and predicted it to be part of a cyclohexene ring and an isopropyl group, which he was correct. However, he tried to experimentally prove there were two double bonds by performing hydrogenation, as we know that an H2 molecule reacted with a metal catalyst under a temperature of 150 degrees should result in an electrophilic hydrogenation reaction. However, that experiment proved indecisive for unknown reasons. but he explained his extensive process of isolating the CBD from a sample of red cannabis oil from a Minnesota wild hemp plant through distillation with ethanol. The isolation was done through more careful distillation processes with 3,5-dinitrobenzenoate. The red oil that was extracted from the Minnesota hemp was distilled with ethanol, which is still greatly practiced and used today. Ethanol is mostly non-polar structure with polar and electronegative OH groups that makes most substances soluble in it, including CBD. For about 20 years after this, there wasn't much research on further expanding the knowledge upon cannabinoids due to certain restrictions from the government, but in 1963, Raphael Mechoulam took more steps towards understanding specific effects of individual cannabinoids by identifying the stereochemistry, or the stereoisomerization of CBD. And with the mention of stereoisomerism, I'd like to take a break from the history for a second, just to look at the structure of the molecule and compare it to THC as it relates to the next discovery in the endocannabinoid system. CBD is made up of carbon, hydrogen, and oxygen, with the chemical formula C21H30O2. It notably contains a benzene ring with 2 hydroxyl groups, or a benzene diol, or a catechol, with an attached pentyl branch, a cyclohexene with an attached methyl group, and an isopropenyl group. The diol and cannabidiol probably comes from benzene diol, as the 2-hydroxide on the benzene ring is what separates CBD from other cannabinoids like THC and CBN that have phenol rings with 1 hydroxide group attached. Alright, so I'm just going to run over the IUPAC name and the isomerism real quick. So I'm going to use WHO's IUPAC name as an example. So right off the bat, you can see that that the pentyl branch right here is marked as 5, so 5 right here, and this is based around the benzene right here, and we see that its 2 hydroxyl groups are on 1 and 3, which means that they probably numbered this like 1, 2, 3, 4, 5, and then 6. And so yeah, we got 5 your pentyl branch right here, benzene 1,3-DL. And as you see right here for 2 right here, they gave it its whole own bracket just to separate it, because it's quite big and hard to describe. So it seems that for right here, the methyl group, they labeled this as 3, and then the double bond right here, 2, and then so it's probably 1, 4, 5, 6, and the 6 right here also in this one, it just represents the isopropanol branch right here. CBD structure is nearly identical to THC's, with the key difference being a cyclic ring formed instead of a second hydroxyl group on the benzene ring. To understand this difference further, we can actually look at a reaction of when CBD can crystallize to THC, as CBD is a crystalline solid based on its structure, under acidic conditions or under pyrolysis, which is thermal decomposition under elevated temperatures for a long time, which causes chemical changes in composition. So a strong electrophile, hydronium, breaks a double bond and a hydrogen bonds to the carbon, similar to the reaction mechanism you'd see for hydration of an alkane. From there, one of the electron lone pairs forms a bond to form a cyclic ring structure, changing the branching structures a bit also. The stereoisomerism of CBD is a bit different from THC too, with a difference in conformational stereoisomerism, as the sigma bond between the benzene diol and the cyclohexene can be rotated upon. Now, these structural differences explain why one is intoxicating and one is not particularly because of some huge differences in chemical properties that exist because of a difference of structure within the structure itself, but of how the different structures create differences in how they interact with the human body, which brings us back to our historic timeline of CBD research and the discovery of the endocannabinoid system. The endocannabinoid system was discovered in 1992, particularly from research that stemmed from the research of cannabinoids in the first place, when research was happening on how they work in the human body. Scientists discovered that the body has its own endocannabinoid system that produces its own endocannabinoids that attach to CB1 or CB2 receptors in the brain. The CB1 or CB2 classification refers to the 2 subcategories of the endocannabinoid system. And as we produce endocannabinoids in our bodies, the cannabinoids that are produced in hemp and cannabis are called phytocannabinoids. Now relax, just because our body has a system that was made to interact with cannabinoids doesn't mean that we were put on this earth and destined to smoke cannabis. I mean, like, nicotine binds to some of these same receptors. For example, the body produces anandamide that releases feelings of pleasure, satisfaction, and appetite. And this is released usually after some sort of athletic activity such as running, which creates the phenomenon we know as runner's high. THC binds to these same receptors in place of anandamide, releasing similar effects but on a much stronger, sustained, and psychoactive level. The way CBD interacts with these structures is different, with the different shape it has compared to THC, both branching-wise and through its stereoisomerism. So as I mentioned, THC has such strong effects as it is very similar to the shape of anandamide and binds perfectly to CB1 receptors. But as CBD has different shape, it cannot bind to CB1 receptors in the same way that THC perfectly mimics anandamide and releases strong effects. But it can bind to other types of endocannabinoid receptors and release different non-psychoactive effects. And not only that, CBD can actually still bind to the CB1 receptors in a different way while THC molecules are still binded to them. And so this actually counters or suppresses some of the effects of THC, which is why many report their experiences of mixing consumed cannabis with both large amounts of THC and CBD to still be psychoactive, but in a more calming and relaxed way, as CBD kind of suppresses the extremely strong effects of THC and just makes for a more calming psychoactive effect. As mentioned, CBD binds to a number of other receptors as well. To start off, serotonin receptors. CBD directly activates a serotonin receptor called 5-HT1, or hydroxytryptamine, which activates an anti-anxiety effect. This receptor is implicated in a range of biological and neurological processes, including but not limited to anxiety, addiction, appetite, sleep, pain perception, nausea, vomiting, that CBD may help alleviate. Next, there are vanillinoid receptors. CBD directly interacts with various ion channels, which are membrane proteins that can act as receptors to create a therapeutic effect. So CBD, for example, binds to TRPV1 receptors, which also function as ion channels. And TRPV1, also known as a vanillinoid receptor, is known to mediate pain perception, inflammation, and regulate body temperature. And finally, we have the orphan receptor, GPR55, which has been dubbed as an orphan receptor because scientists are still not sure if it belongs to a larger family of receptors. So it is quite unique, but GPR55 is widely expressed in the brain, especially in the cerebellum. GPR55 promotes an osteoclast cell function, which is mediating and fighting against bone damage, which facilitates bone reabsorption. Thus, this type of cell function can be seen aiding in the spreads of certain types of cancer, but CBD happens to be anti-GPR55, which means that it can suppress this receptor, decreasing cancer cell proliferation. But unfortunately, that would also suppress the bone regeneration to a certain extent. Similarly, CBD also exerts an anti-cancer effect by activating nuclear receptors in your body, PPARs, peroxide proliferator activator receptors, that are simulated on the surface of cells' nucleuses. PPAR gamma that is released from these PPARs can be linked to the development of Alzheimer's disease and a number of other diseases, but CBD once again suppresses PPAR gamma and reduces these effects. Additionally, a 2015 paper by MPDI described THC and CBD's antioxidant potential, meaning the ability to clean up free radicals left behind in the body, as they can damage the body and are linked to cancer among other diseases. THC, however, was described as a better antioxidant, as with higher electron donating abilities and its phenol group that regularly possesses good antioxidation throughout most of the molecules they exist in. As per the biosynthesis pathway of CBD, it starts off with cannabidiolic acid, CBGA, in the hemp or cannabis plants. It then goes through a process called cannabidiolic acid synthase, where CBGA reacts with oxygen that is available to it, as it's a plant, and produces cannabidiolic acid, CBDA, with the byproduct of H2O2. And a fun fact, cannabidiolic acid synthase is actually being studied and attempted to be cloned because the process is tied to a hemp plant's growth, which could relate to natural fiber production. And raw CBDA has many of its own medical properties similar to CBD, but slightly different that have yet to be studied more extensively as well. CBDA is then converted into CBD under decarboxylation, under 105 degrees heat reacted with CO2 to remove a carboxyl group and replace it with a hydrogen. The reaction mechanism involved a hydrogen breaking away from the bond with the oxygen in the hydroxyl of the carboxyl group, causing the double bonds to move around and the hydrogen to bond with another oxygen, creating an enol. H3O acid then reacts with the enol to form a ketone and form CBD. The mystical and not fully comprehensible way CBD interacts in specific ways with receptors in the endocannabinoid system to create positive effects and suppress negative effects in our body is extremely interesting, and it may repel some patients away in its mystery and unexplainability, and it may attract others because of its mystic. Or perhaps this molecule is surrounded by uncertainty because of the lack of attention to it. I had to throw myself into deep research of giant medical and chemistry journals and paper databases to find a lot of this information, as the plant that it comes from was slandered in controversy and critique in the 20th century. But meanwhile, CBD and its healing properties were waiting there for the medical world to explore, and it still is.